Hello, so we are Tapton School from Sheffield, and we're the Northern Hub for the Institute of Research in Schools. And today we're going to be sharing with you our exciting new project, the molecular mechanisms behind motor neuron disease. So, over the last 4 years, we've been researching the role of novel genes in cardiovascular disease, and the generous funding from the Wellcome Trust means that we can carry out many molecular biology techniques. The Sheffield Institute for Translational Neuroscience is a world-leading research facility, and it's dedicated to alleviating the effects of neurodegenerative diseases. And unlike many other research facilities, it combines scientists and clinicians to accelerate research findings and into medical advances and treatments. And our collaborator, Guillaume Hartberg, who is the head of the RNA laboratory at Citran, has supported us for many years. And we will be talking about his research findings and his most recent publication, which is just there, and what science holds in the future for this area. And I was lucky enough to spend 4 weeks in his laboratory doing a Nuffield research placement, and this was in the summer holidays, and I'll be presenting my research from that.
Motor neuron disease is a debilitating neurodegenerative disease. It results in initial loss of muscle strength in the limbs, and eventually the disease takes over all motor functions in the body, resulting in progressive paralysis. Patients will require life support to ventilate their lungs before death. In most cases, the disease is rapid, where most patients die within 3 to 5 years of their first diagnosis. This was the case with Lou Gehrig, an American baseball player who you can see on the bottom right, who had famously the highest average home run score before succumbing to this terrible condition. In America, motor neuron disease is sometimes referred to as Lou Gehrig disease, but now more commonly is known as ALS, Amyotrophic Lateral Sclerosis. This disease is, however, complex. As in the case of Stephen Hawking, who is on the top right, he has had motor neuron disease for over 5 decades. The disease can be inherited, familial, or sporadic, resulting from mutation. Its onset is from the age of 50 and can be found in younger patients as well. Its incidence is relatively high, with 1 in 400 people contracting this disease. With an ageing population and advances in cardiovascular medicine and cancer, the impact of this disease is clearly increasing.
So I'm going to talk for a little bit about the background to motor neuron disease, and more specifically looking at the central nervous system and the brain. So, if you couldn't tell, that's a brain on the board, and in the centre here, in the middle of the cerebral hemisphere, we have the motor cortex. Now, the motor cortex is involved in movement. So, if we have a look here, we have a section of the motor cortex. We've got this folded tissue around the outside of the brain. And you can also see that different sections of the motor cortex control different parts of the body. So, for example, if we look at a stroke victim, this can explain why they can have a variety of different symptoms. So, for example, let's say they have a ruptured blood vessel here around this left-hand centre, then that can result in the characteristic drooping of the face, whereas if there's a ruptured blood vessel, let's say, at the top of the brain here, that could result in loss of movement in the arms. So I'm going to look a bit about motor neurons now. So there are upper motor neurons that connect the brain to the spinal cord, and then lower motor neurons that connect the spinal cord to muscles. Now, motor neurons are very long cells, which you've probably seen this diagram on the right before. They've got that characteristic cell body with branched endings and then a very long insulated axon. Now, to put the axon to put the whole cell into context, if you, let's say, scale the cell body to the size of a golf ball, the axon would be as long as the Empire State Building is tall. And I don't think you need a scientist to tell you that that's quite big. So now to have a look at the cells of the brain. So the motor neurons are the focus of this, of our studies, as obviously these are the cells that degenerate during motor neuron disease and how this, and as this disease progresses. So on this diagram on the left here, the motor neuron here is shown in green, but we also have another type of cell called the astrocyte, which is shown in red. And these help connect the motor neuron to the blood vessels, as well as providing metabolites and all sorts of stuff. So along with metabolites, the interactions between astrocytes and motor neurons are plentiful, as you can see. There's all sorts of things going on. So it's important that we understand these interactions in order to gain an understanding of how motor neuron disease works. So one hypothesis suggests that the astrocytes provide too much neurotransmitter, and this causes motor neuron cells to be overstimulated, and therefore the environment becomes toxic, and thus the cell can become damaged and die. However, there are many, many possibilities on what could cause motor neuron disease, which is what makes it a very complicated disease to try and treat.
So this slide shows a timeline of the discovery of genes involved in MND. So in the mid-90s, a free radical scavenging gene, SOD1, was identified as a causal factor. So there's a lot of research ongoing into this mechanism, and Citran is involved in pioneering treatment into this aspect of the disease. However, there are many other genes that are involved, and lots of these are involved in the control of gene expression. So this figure uses dots to represent publications. So the bigger the dot, the more publications there are. So this yellow dot is C9ORF72, which is a key gene in this disease, and it's the subject of our investigations.
So C9ORF72 is not the best gene name that we have come across. The reason being that this function has yet to be characterised. This name therefore simply tells us that it is on chromosome 9 and it's in the 72nd open reading frame on this chromosome. The gene has been sequenced, and this figure shows that it consists of 11 exons. The most interesting aspect of this gene is the non-coding regions. So in the first intron, there's a hexonucleotide repeat, GGGGCC. In patients with MND, this repeat can be up to 1,000 to 2,000 repeats long, whereas in non-MND patients, it's only repeated 3 to 6 times. Over 30 repeats is considered as pathogenic.
So there are two main proposed mechanisms for the toxicity of these repeat sequences. The first mechanism suggests these multiple repeats bind to important regulatory proteins in the nucleus, eventually leading to the prevention of normal cellular function. The second mechanism suggests these repeat sequences actually exit the nucleus and are translated into proteins, dipeptide repeats. These DPRs are not physiological and therefore would aggregate in a cytotoxic manner, resulting in a common problem. In using a fruit fly model, cytotoxicity can be monitored by looking at the development of the compound eye. This is because the production of the dipeptide repeats in the fly disrupts eye development, as shown on the right here. So how is this happening? In AS, you learn about the transcription and translation, and then go on to learn how gene expression is regulated in A-level. But still, these are oversimplifications. We quite happily accept that pre-mRNA is transcribed, and that this is then processed by splicing, which results in mature mRNA leaving the nucleus for translation here. The level of regulation that is responsible for the protein expression is mind-boggling, as this diagram demonstrates. But essentially, many different proteins are involved in the export of RNA from the nucleus. Now, the protein that we want to focus on is called SRSF1. It's a nuclear export factor, which has been previously reported to be an important motor neuron disease. RNA is indicated here in red, bound to SRSF1, shown in green.
So what do these results show? All right. So in this diagram, the hypothesis suggests that the expanded intron sequence of the C9ORF72 binds to the nuclear export factor SRSF1, which you can sort of see there. This binding facilitates the nuclear export of the unspliced version of the messenger RNA. RAN translation, which is translation without a start codon, then results in the production of dipeptide repeat, which would have a toxic neurodegenerative effect. So to test this, Guillaume and his collaborators suppressed the expression of the SRSF1 protein in flies, which had C9ORF72, with increased repeat sequences within intron 1. As you can see, when SRSF1 is reduced, the toxic effects of the repeat sequences are massively reduced. This was further supported with assays of fly and larval movements in climbing assays, where the negative geotaxis response is determined by allowing Drosophila to climb a measuring cylinder. So you can see that measuring cylinder there, and basically when the flies are affected by motor neuron, they struggle to climb it, so you can time how long it takes them to climb the measuring cylinder. So this diagram shows that in the absence of the SRSF1 nuclear export factor, the unspliced RNA cannot leave the nucleus, and thus there is no translation of the dipeptide repeats. So by reducing SRSF1 levels in motor neurons from ALS patients, the toxic effects of astrocytes mentioned earlier are alleviated. Here you can see motor neurons, in the top left, you can see motor neurons, in green growing over a surface of astrocytes, in the control but inhibited in the ALS, so that's the bottom left. Suppression of SRSF1 allows the motor neurons to develop normally, as seen on the right side.
When doing this research, it is important to also check that the dipeptide repeats are being expressed. Here we are showing the DNA constructs used to produce dipeptide repeats RNA. This is then transfected into cultured cells, and the presence of dipeptide repeats is detected using Western blot. Essentially, all the proteins are extracted and separated by gel electrophoresis, which is essentially chromatography, as you can see. The proteins separate by size, and the specific antibodies are used to detect the presence of proteins. This shows the expression of the DPRs in cells given the DNA construct, as well as the reduced amounts when SRSF1 nuclear export protein is reduced.
Our experiments will perform similar assays, but instead we'll be using the yeast model organism. This is unusual, as yeast is a single-celled organism with no nervous system. However, yeast cells are eukaryotic and have a nucleus, and therefore have many of the same nuclear export mechanisms. Our aim is to transform yeast with a DNA construct that contains the repeat sequences from C9ORF72, so that we can further investigate the other nuclear export factors involved. The diagram in the middle shows the first step, which is to clone the DNA from Guillaume's constructs into a yeast expression vector, so that hopefully the yeast cells will make the dipeptide repeats, and then we will analyse by Western blot to see if they are present. Previous publications have shown that the dipeptide repeats demonstrate a decreased growth phenotype when expressed in yeast, so this should provide an assay for determining whether the dipeptide repeat phenotype can be rescued in the absence of nuclear export factors. Thank you, and we'll now answer any questions.
Wow. Hello. I'm in awe of you guys. not only the level of the science, but also the presentation skills, which are awesome. Let's bring you forwards to the front so that they can ask you some questions. Come on over. Do we have any questions in the room? Go on, who wants to throw a question their way? Can be about the whole process, what they enjoyed, any problems they had along the way. Okay, I'll start. What was the one kind of thing in this whole process that's kind of stuck with you that you'll be taking forwards? So that could be a problem that you had to overcome, or is it just being involved in the first place? Like, what really got you?
It's kind of just...
I'm coming to you with a mic.
I'd say just becoming familiar with the disease and how horrible it is, and just seeing the effects of it. It's been quite traumatic in a way, just seeing how it's affected the people, the family of the patients, and just how it's pretty much kind of stopped people living their normal lives. Yeah.
But do you get some sense of hope with this research?
Yeah, so our research is actually quite promising. So the research that I was talking about, which I did with Guillaume, is really promising actually. It's got a lot of results that show that it might be able to be moved to the next stage of drug therapy. And because we're working with patients, then it means that it can be accelerated because the patients are right there, so we can test the drugs on them if they're ready.
Cool. Well, nice one. We will watch. Brilliant. We have a question from a recognisable face there.
The diagram you had of the affected motor neuron showing the myelin sheath being degraded. Is in motor neuron disease? Is it specifically motor neuron? I mean, the name implies it is, but I mean, is there any damage to sensory neurons as well in terms of myelin sheath and demyelination? And what do we know causes that demyelination or triggers the demyelination in MND?
I volunteered.
It's possible that there is a potential that could happen. However, our research is specifically focusing on different export factors in the nucleus, and specifically the potential random translation of these GGG, GGC sequences that create the dipeptide repeats, and that's really the core focus of our research and what we hope to look into further.
Brilliant. Right, give them a huge round of applause.